Yeah. So good afternoon, everyone. I will talk a bit about our journey in search of climate resilient vanilla species. So it is an ongoing project with several institutions from Costa Rica, Belgium, Mexico, Colombia, the United States. So as it is an ongoing project, I will present the main purpose, the approach, and some very preliminary results. Okay, so I think we're all aware of the fact that climate change is affecting several sectors of our society, and especially the agricultural sector is expected to be severely affected. So, jeopardizing food production and thus food security. And especially the tropical regions are expected to be quite vulnerable. And an example of a tropical crop that may suffer under climate change is vanilla. So vanilla is a high-value cash crop for many smallholder farmers in the tropics and a globally appreciated spice that is used in several industries. So here on this picture, you can see typical vanilla plantation in the south of Costa Rica using living support trees. So why do we think that the crop species or why do we think that vanilla may become vulnerable in the future? Well, because several studies have indicated that the genetic diversity within the cultivated lineages of the crop species, which is mainly Vanilla planifolia, is rather limited. So this is due to the fact that most of the cultivated plants are descending from just a few, let's say, mother plants that were propagated in a vegetative way. So this has led to what we call genetic erosion. So plants that are characterized by more susceptibility to biotic and abiotic stresses. And these stresses are expected to increase under climate change. So that's why it might be that the supply of our beloved spice may become at risk. So there is a need to enhance the resilience of the vanilla sector, and this actually at several sections along the supply chain. And one way to do this, especially at the production side, is through diversification. So diversification at system level by implementing more robust agroforestry systems, or at crop level by looking at the possibility to integrate climate resilient vanilla genotypes. But where can we find these? So there exist what we call crop wild relatives, which are species closely related to the crop species. But being wild species, their populations can be found in natural habitats. So they're actually continuously evolving and adapting to different and changing environments. So thereby developing traits that might be of interest for crop improvement and breeding. So in the case of vanilla, we're mainly interested in primary and secondary crop wild relatives. So the primary or the wild species of Vanilla planifolia, so the wild populations, sorry, and the secondary gene pool are the or other species that are closely related to Vanilla planifolia that are actually found within the section Sanata. And these are actually species native to the neotropics and that produce fruits that contain vanillin or other vanillin related aromatic compounds, so that may be of interest for the industry. So despite their promising role in in-depth evaluation and actually action plan to integrate novel genetics into crop breeding, especially focusing on the one with potential climate change resilience traits, is currently lacking. So we need more information about intra and interspecific diversity, especially of these wild populations, and to look for actually identify species or populations with high adaptive capacity and see the possibility to incorporate them in crop breeding. Um, so this information can help us to formulate what we call provenancing strategies. So actually look at, okay, which populations can serve as donor populations for use in crop breeding. But at the same time, we also need to develop conservation strategies because lots of these species and populations are actually endangered. So if we want to enable the use of them, we simultaneously need to develop concrete conservation schemes. So the aim of this project is to identify potential climate change resilient vanilla species and populations to formulate these provenancing and conservation schemes. And how are we going to do that? Well, by defining the vulnerability of these species and populations. So vulnerability to climate change is actually defined through 3 dimensions. So you have exposure, sensitivity, and adaptive capacity. Exposure and sensitivity refers to the degree to which an area may become exposed to climate change and how this will affect the species that occur there. So actually how it will affect the suitability of the species to occur in this area. So this together, exposure and sensitivity, defines the ecological vulnerability of your species. Then you have the adaptive capacity, so the capacity of a species to adapt to changing environments. And this depends more on the genomic composition of your populations, and this is what we refer to the genomic vulnerability. And these two together are actually defining the overall vulnerability and will help us to gain insights to provide these provenancing and conservation schemes. So the approach, well, first of all, we need to define a target area and target species. So like I said, we are mainly interested in the primary and the secondary gene pool vanilla crop wild relatives, which are native to the neotropics. So to assess the ecological vulnerability, we will use species distribution models, and for that we will take into account the full distribution range of these species. So we will focus on the neotropics. Then for the genomic vulnerability, we will use genotyping by sequencing techniques, and for that we are focusing on 3 countries. Sorry, 3 countries for now: Mexico, Costa Rica, and Colombia, which actually represent the north, the center, and the south, more or less, of this distribution range of our 8 target species. So this is the target area and the target species here. We're focusing on 8 for now. They're listed here, and most of them occur in the 3 countries that we are focusing on for our GBS. So the first part of a study is to assess the ecological vulnerability. So we will do this by implementing species distribution models using the Maxent algorithm. I won't go into detail, but this algorithm has shown to function very well or quite well with rather limited occurrence data and also sampling bias. So what do we need to perform these SDMs is occurrence locations, of course, of our species. So you can gather them through online databases, own inventories, literature, and so on. Once you collect these occurrence data, of course, you need to filter them because there are quite some errors, especially from online databases, so that you have a final data set with at least less errors to use for your SDMs. Then you also need environmental data, so data on climate, topography, soil, and so on. So abiotic variables that actually define the suitability of a species to occur in a certain area. You can also include biotic variables such as pollinator interactions or interactions with seed dispersers to have more complete models. But that's something that we're doing in a parallel study. So for now, we're focusing on including the abiotic variables. Once you have done your models, you get on the left what we call suitability maps. So actually maps showing the suitability of a species to occur within the area that you modeled. So ranging values from 0 to 1, with 1 being highly suitable to occur within this area. And then you can transform these maps into presence-absence maps by using a certain suitability threshold under which a species will probably not be present. So that's what you see on the right. So once we have these models for our current data, we can then project them to the future because there are different scenarios and climate models available that you can use to project your models to the future. So on the right, I won't go into detail, but there are several general circulation models or climate models that were developed by several research institutions across the world. So depending on the target area, so the area that you want to model, you can select the best models, so the best climate models, GCMs. So that's what we did. We selected 5 using the scheme. And then we also selected 2 climate change scenarios. So these are the shared socioeconomic pathways. So these are actually different scenarios of socioeconomic development that have an effect on greenhouse gas emissions. So you can select from different scenarios, going from very positive to very very pessimistic. So we choose one that is rather realistic and then one that is a bit more pessimistic to see how suitability of our species will change under these different climate change scenarios. So once we have done that, so our models for our current data and our future data with these different scenarios, we can then actually extract the suitability maps to calculate the ecological vulnerability. So the more or the less suitable a species will become in the future, the higher its ecological vulnerability. Then in the second part, we want to assess the genomic vulnerability. So for that, we need to explore the genetic diversity present among the wild populations of these vanilla species to see if there might be some adaptive capacity present. So for that, we need to go into the field and collect plant material. So we are collecting leaves, preferably young leaves, that we extract the DNA using the CTAP method that was adapted for vanilla, and then sequencing using diversity array technologies. Once we have our sequences, we can then identify SNPs, so to look for differences among our samples that we can use to create phylogenetic trees and to better understand population structure. But more importantly, or what we are more interested in, is to relate these SNPs with environmental data. So to look for correlations between specific SNPs and climate data. And we will use redundancy analysis. So this is actually analysis that allows you to correlate your SNPs with different environmental variables at the same time. So what we are actually looking for, or you can see here on this histogram, so it's presenting the frequency of your SNPs on like the one of the RDA axes of your analysis of your RDA analysis. So in the center, these are what we call neutral SNPs or random SNPs, so they don't show any correlation with environmental data. So we are mostly interested to define these ones, the outliers, because these are correlated with specific climate variables. So for example, with drier environments or with environments that are characterized by higher precipitations. And once we know that for our current climate data, we can also project this to the future and see actually how much change there would be needed for a population to cope. So how much change in this allelic composition would be needed to actually cope with the future climate conditions. So in this way, we can define the genomic vulnerability because the more change is needed, the higher the genomic vulnerability. So the lower its adaptive capacity to climate change. Then once we know the ecological and genomic vulnerability, we can actually combine them to gain more insights into, okay, which populations can serve as donor populations and which populations may need more attention regarding conservation. So for example, populations with high genomic vulnerability and high ecological vulnerability, they may, they may need actually ex-situ conservation because their habitat won't be suitable anymore and they're not having the capacity to cope with these changes. So these might need ex-situ conservation. Then you have on the other side populations with low genomic and ecological vulnerability. So these these are the least vulnerable, so important to conserve in situ. And these populations may also serve as donor populations for crop breeding programs. So this is a diagram that we actually adapted a bit from Dawson. So to eventually the idea is to plot so all our populations on this diagram to see which populations belong to these different categories. So some very preliminary results, as I said, because it's an ongoing project. So what we did so far was carrying out suitability distribution SDMs. Sorry. So the models resulted to work out very well. So the AUC values were quite good. 7 out of the 8 of our target species could be modeled. This is due to the fact that one didn't have enough occurrence data, which is a common problem to perform SDMs, because you need at least 30 occurrence data points. But what we saw is that all of the species that we could model had a decrease in habitat suitability by 2050, and this were the two scenarios that we modeled. But there was some variation among species, and a general trend was that the suitability was the same or actually increased to higher elevation areas. So this is what we saw. It was a common trend among the species. So this is then when I showed you before. So if you have the suitability map of your current data and the suitability map of your future data, you can actually create, so overlay them to see the genomic vulnerability. So this is, for example, for planifolia. So, oh, sorry. So the green areas show that they remain suitable or even may become more suitable, and the red areas mean that the areas will become less suitable. So the little black dots, although you cannot see them very well and it's a bit confusing with the islands, but the little black dots are the populations. So you can then see from these maps which populations are laying in these different areas, so the less suitable or the more suitable. So we did this for all our target species, well, at least 7 out of the 8. I think I need to hurry up a little bit. So this is for the ecological vulnerability. Then the genomic vulnerability. Oh, so what I forgot to say is, so if you have, for example, a population now in a red area, so it means that the ecological vulnerability is high. But if this population shows a low genomic vulnerability, sorry, it may actually be possible for this population to survive in this area. So this is why it's important, the next step, to assess the genomic vulnerability. But this is still work in progress. So actually, we finished our data collection in Costa Rica. This data is now being processed, but we still need to collect our data in Colombia and in Mexico to have our final data set that we can then use to assess the genomic vulnerability and combine it with the ecological vulnerability. So yeah, the expected outcomes of this project is that we can use this information to actually formulate a conservation scheme for these target species within our target area, so the neotropics, and also that we can identify potential populations with high adaptive capacity that we can use in provenancing, so for provenancing. And this is actually the first part of our research project to actually do a screenage, because once we can identify these potential donor populations, we can then implement greenhouse experiments to actually see, okay, if these populations, so based on our first screenage, actually show resistance against drought, for example, or against weather conditions, higher precipitation conditions, or higher temperature conditions. So this is the first part of the project, and then the second part will be the greenhouse experiments, and in the last part will actually be the field trials to work with the farmer communities to see how we can integrate these identified phenotypes into the real world, let's say. So yeah, thank you very much, and I'm happy to answer any questions and suggestions also.
Thank you very much, and we have time for a few questions.
Hi, I really love your project. Really good talk. I was wondering if there had been any germination experiments or in vitro lab trials with the crop mold relatives that you've looked at already, so that you might be able to use that to kind of ground truth some of the models you're using with Maxent.
You refer to germination experiments?
Yes, or any other kind
Uh-huh.
of in vitro growth trials, field trials,
Ah, okay,
something
that
like
we
that.
can use like as a base.
Yes, yeah.
There are some in vitro experiments that have been done, but this project is focusing on extracting material from wild populations, and the in vitro experiments that I'm aware of that have been done is with material from living collections at research institutions. So we are actually more interested in going into the field and looking at at these natural populations at current conditions. Is there something interesting that we can use for crop breeding? So no, at present I don't think there is like from wild populations in vitro plants available that I know of. Adam?
Thank you.
Thank you very much. Do we have other question?
Thank you for your talk and for the methods that you explained. Very interesting how you addressing this question. And I'm curious about this models. How can we add to the final models the idea of the loss of environments? Because yeah, you have a projected area that can be occupied by the species based on climate, soil, whatever. But then that's not a suitable area really, because then you have all this loss of natural places, right, for living. So I'm just curious. Maybe you have think about it or not, but it would be interesting. I don't know if it is a way to model the areas that can be lost, maybe to 2030 or 2050. Project this in years and see. Yes, we have this suitability, but this is actually not, because we have areas that are taken for agriculture or other uses.
Yes, that's a very good question, and actually, it would be very good to integrate this also as a variable. So you can do this because you can actually create certain scenarios that you say, okay, maybe positive, so there won't be any destruction, and then different levels. So to okay, it will be complete destruction. I don't know. You can create different scenarios and integrate this in your models. It's actually a good observation. Thank you.
Do we have any more questions?
Thank you for a very interesting project. Can you comment a little bit more about how you will do the exposure experiment? You will be put plants in control conditions, and do you plan also to see how what is the impact on the flowering of the vanilla?
Yes, if we have time, that would be nice. Yes, so it will be in a controlled environment where we will simulate droughts, increases in temperature, so different abiotic stresses, and see how it will influence plant growth, vitality, and hopefully we can get to the flowering and the yield, and because that takes 3 to 4 years more or less before they start flowering.
Thank you very much.